Thank you for coming. Thank you for the opportunity to talk with you today. I'm going to take a little bit slightly different approach. Doug is, I've been following Doug's work for probably 10 or 12 years now, and it's the assay guidance manual I'm going to reference actually in a lot of my slides. It's that informative, and you all should be able to follow that information. But what I thought I would do is I took the approach to try to address some of the questions that usually come up in the lab when we start trying to work with somebody on a project. And so that was sort of the approach I took. I'm going to talk a little bit about what the core has, what we offer, how we go about approaching a new project, and then if time permits, I've got a couple of projects at the end I thought we might just kind of walk through to kind of give you an example of what it is. But I also want to emphasize that we are a collaborative resource. This is not where you drop off your project, we do a bunch of screening and give you some results back. It is truly a collaboration between your laboratory and ours, and we encourage and want people from your laboratory working with our staff and actually working on your projects. So personalized medicine or precision medicine has been in vogue for now quite a while, and it's sort of the goal that we're all trying to attain. And that's really moving the things right from the laboratory to the patient. And this is sort of the stylized version of that. And the whole idea is to try to get the right dose to the right drug to the right person at the right time. But unfortunately, this is kind of where we are right now. There's still this large gap between the laboratory bench and the patient. And we're hopefully going to try to show there are instances where the screening that we do might help bridge some of those gaps. And that's really what the core is trying to do and what we're trying to accomplish. Doug obviously showed that drug discovery, it can be a very complex process, and that's illustrated in here. Trying to get from your idea, your hypothesis here to maybe to develop a new therapeutic approved by the FDA is a very long and arduous process. There are a lot of circles and loopbacks, a lot of areas for success and for failure. This is the more stylized view that we might notice. But you can notice it takes a long time, it takes a lot of money to get there. High-throughput screening, what we do, tends to focus more in this area right in here, which is moving from target validation to identify active compounds, validate those compounds into leads, and then move those, hopefully those will move on towards clinical trials and move into the approval process. But we tend to stay focused down in this area down here, which bridges the gap between where you guys usually are on developing new targets and new validated models. And then we're trying to move those through into something that is clinically translatable. In doing so, you have to realize, though, that when we're working with things and we're trying to screen large numbers of samples, the immediate human relevance, as you can see in this chart, is low. I mean, we're trying to screen lots of purified proteins and cells and culture and things like that with large numbers of compounds, and that's the high throughput part. But as you move towards the relevance towards human, you can see that those numbers dwindle quite quickly, and it's what you're able to do. I mean, just think about an animal study, trying to study a drug in an animal, maybe a minimum of 10 animals for a drug, and then plus controls. And I mean, you can see how the numbers would escalate fairly quick, and you can't do 100 compounds that way. It's just not possible. So how do you get from that 100 down to a manageable number? And that's where I hope that we can make presentations. There are basically 3 ways to develop new drugs. You can actually develop a new chemical entity. That's something that's new and novel, not been designed before. That's what a lot of people ask me if we can. It's not something we do a lot of. And you'll see as we get into this probably some reasons why we don't do it, but there's ways to do it through collaborations with the GCC, and I think that's important. Repurposing is something that we focus on a lot, and that's because most of our funding comes from CPRIT, so we're obviously trying to do translational projects. So probably 80% of the projects that come through the lab are cancer in nature, and we're trying to look for translational projects as best we can. Or, in the third way, is just to take drugs that are new, reformulate, and try to use new ways to do that. Again, our model here, our repurposing set here, really what you're doing is you're taking approved drugs that are already known, they have toxic profiles, so this part's usually known, and we can kind of skip through that and try to get from activities here to phase trials, but it takes a lot of work still to get there. I was talking with a colleague last week who works in GBM, and he says, well, it's nice that you can do all this work, but unless it's done in a CLIA certified lab, I really can't translate it directly to a patient. So there's still obstacles and roadblocks, but that. We're trying to address those. That pathway is illustrated. This came out of the assay guidance manual. This is a table that they had. I thought it was kind of interesting to see. You know, if you're going from target identification from the bench towards the patient, which is as you're going down, these are milestones that you would try to accomplish as you go down that way. And you can see sort of estimated costs. These are from, I think, 2014 based costs. You can see how that escalates, and then you can see sort of the timeline that you're looking at. So just getting from the bench to a phase 1 trial, you're looking at probably in the range of 6 or 7 years, and then to try to get it into a phase 2 trial, now you're starting to look at 8 years, 9 years, and now you can see how the millions are multiplying. But now the estimate is about $2 billion to actually develop a new drug. So just a couple of things then. Now, trying to move from where you are, target identification, towards screening. So I thought I would start with that. So really, to start moving towards that, you obviously want to make sure that the target that you're identifying plays some role in the disease. And so that's either something that's been published in the literature, maybe new biology that you're developing in your group, and that's what we're trying to focus on. And so there's a couple of ways to do that. I mean, you have a target that you've identified, and you say, well, I need a drug, and so, or a probe or some molecule. So you want a drug, and so you're working this way. The other way to go is you have a compound, and you want to know what it does, so you can work on a phenotypic assay and try to work that way and try to do target validation. So a couple of different ways to do that. You can do de novo, where you're trying to develop a new, against your particular target that you've identified, or you might have a drug that you've seen an effect, but you're trying to figure out how it's working. And we can work from that way as well. And so then some of the other desired properties, obviously, can the target be screened by, can you develop an assay for it? Can we actually do a high throughput screen? And this one's kind of interesting. I thought it was kind of nice that it's not expressed throughout the whole body. And why would that be? Why would you not want your target to be expressed everywhere? Anybody want to venture a guess? Because obviously if you're targeting it for inhibition, you don't want to inhibit it all the way through the body. It might have adverse effects. So adverse effects can be a problem. And again, these are all slides from the assay guidance manual. Target validation requires demonstration that the target's actually involved in the disease process. And these are some of the criteria that you might try to meet as you're moving to decide whether you want to take your target and move it towards drug therapeutics discovery. And these are some of the ones that come into play with us. Is can you modulate it through genetics? And CRISPR-Cas has been a big player in that now. But this is one of the biggest ones for me. For when we're trying to do screening, is can you come up with a secondary assay that actually points back to the biology? We can do high throughput screens, we can find active compounds, but that doesn't necessarily mean that it's modulating the biology that you're interested in. And so you might need something that's not quite high throughput, something that's more related to the biology. And so we ask you to actually start thinking about developing your secondary screens before we even do the primary screen. So think about how you're going to relate these hits back to your biology before we even get started. Then some of the other ones that you can come up with, are there already known molecules? Does the target have a history of success? And there are many that have done that. Those are called druggable targets. For the academic side, intellectual property. Is there intellectual property associated with your target, or developing probes or new molecules for that target? It's something we don't think about much in our laboratories, but it's very important. Think about whether you have intellectual property in what you're doing. And is there a potential, is there a market potential? That's minor for us at this point because we're so far removed, but it's something to think about and consider. Talking about druggable targets, this is a slide that I took from a review, and you can see that 75% of all targets, and this was long ago. I mean, we're moving well away from this now, but at one time, 75% of all the targets were either a kinase, a GPCR, or a protease. But through as research is going on, we're moving far away from these now. And in the academic side, and the screening that we do in our laboratory, I haven't done a screen against a GPCR in 8 years. It's just targets that we don't work on. We work on unusual and more novel targets than these. So these are ones that we rarely work on anymore. But they were always the druggable targets at one time, especially in the large pharmaceutical companies. So what is a high throughput screen? I just put this up as a comparison between what you might do at the bench and what you would do in a high throughput screen. And really what you want to think about a high-throughput screen as a simple assay. You want it to be fast, you want it to be inexpensive, you want it to be quick. And so what are you doing? You're trying to limit how many steps that you do. I mean, you can, when you're working on your bench, you can do any number of steps. It's easy to do. But when you're trying to automate, and you saw some of the robotics in Doug's presentation, and I must say that we don't have any big arms in our lab, but we do have automation. What are you working on? Are you working in a test tube or a slide? Well, we're working in multi-well plates. And so, you can see the direct comparisons about, but really what we're trying to do is you're trying to limit everything down to really what you want to change in the assay is the compound. You want to see the effect of the compound in the assay. So that's what you're trying to do. But you're also looking for a rapid turnaround time. These are high throughput, so you don't want to take a week to generate your endpoint. You want to be able to get it in a fairly good quarter. So as you screen, you will develop the compounds that give you a response that goes with your assay would be considered an active compound. So then you try to satisfy a couple of criteria to say whether this active compound or not. So to do the screen, can you get the reagents? And that's one of the biggest things. So when we're working with screening, and you come and say, we'd like to do a high-throughput screen, one of the things we're going to say is, can you do that for a long period of time? It might be weeks, it might be months. We've had some projects that have run years. So it just depends on the nature of the assay that you're running. Can we actually develop an assay in our laboratory? So there are some techniques we can't do, and so that's one of the things we have to evaluate. Do we have a library that you're interested in that we can screen? Do you have a criteria for what you would identify as an active compound? So it's something to think about as you're developing your target and you want to do screening is, what are you going to call an active compound? How are you going to identify those? And so those are things that we want to do. So this is sort of a schematic. Again, this is sort of where we are from the bench, and we're trying to get into screening. You can do virtual screening, and a lot of people do. If you have a target that has a homology model, or if you have a crystal structure, you can actually do docking of drug structures against that. It's not something we do, but there are core facilities probably in the GCC that can help you do that. It's not something that we do. We actually do the high throughput screening. We're on this side over here. So we can plug in your virtual screen. We can move, if you want to provide compounds, we're happy to screen your compounds as well as we have our own collections of molecules, and I'll show you those in a few slides. The different ways that we do screens, we can do a quantitative high throughput screen. So if you want to do several concentrations in the primary screen, we can do that. We can screen with single concentrations. Repurposing, again, is our focus. We also work on drug combinations, so we do a lot of combination screening as well. In cancer, for example, if you have a driver mutation or you have a standard of care, you have a resistant cell line, we can actually do standard of care plus something else, something that we focus on. The types of assays we tend to get are biochemical assays, where you're working with purified enzymes, receptors and ion channels, or cell-based. And this is a lot of the cancer models that we get are cell-based models that have reporters engineered. We get a lot of patient-derived xenografts, just from ways we can do those kinds of screens. And then obviously we're trying to identify compounds that modulate the activity. So every assay that comes in goes through, this is sort of the basic workflow. So the first thing is the design. What is your model? What are the endpoints? Then we're going to try to optimize, working with you, to try to optimize the assay towards a screen. We're going to do the primary screen where we're actually screening compounds and trying to determine active molecules. And you want to validate those actives. You want to eliminate false positives, false negatives. And you can do that through either orthogonal or counter screens. And eventually get back to your secondary screen. So that's why we want you to think about it up front, because it's not too far, removed from once you've done the primary screen and validate some active compounds. You're going to want to actually test these to make sure the biology is right. What you don't want to do is get a lot of hits and go down the wrong road, chasing a molecule that's non-specific for your target. So optimization, that involves the miniaturization. So this is where we're starting to work towards the primary screen. So you may have a bench-type assay that you might be doing in a multi-well plate, you might be doing in a strip well, you might be doing it in tube. But we've got to get it into a multi-well plate that we're going to work on. And some of the things that we're going to look at is uniformity across the plate, edge effects, and then some of the statistics we look at. I'm going to show you one that we use a lot, Z prime. I don't know if everybody's familiar with it. I thought I'd put it in there. And then variance, confidence variance, and then we have to try to automate the whole thing. Because remember, we're going to try to screen hundreds or thousands of compounds. We're not going to do that by hand pipette. We're actually going to use the automation to do it. So this is one example. This is a cell-based assay where we're just DAPI staining cells and counting them. We plate the cells. Next day we do a stain on those, and then we let them grow for 3 days and just look at the growth between the two. And I just used it as an example to try to show some of the statistics that we look at when we're trying to do performance optimization. So the variance in the data between the controls is fairly tight. Some of the ones you might be familiar with are signal to background, where you're looking at just the max over the min. You can see we've got a signal to background of about 4. If you want to take into account signal to noise, so here, this doesn't account for any of the variance. At least here we're looking at the variance in our low sample. As you can see, we have a fairly tight bit of data here, so we've got a very high value for that. So you can see from these, this is sort of the statistics that go with that. The Z prime, however, the nice thing about it is it's taking in not just the variance of the minimum sample, the low end, but also the maximum. And so really what you're looking at is sort of the 99% confidence interval around each mean for the low sample and the high sample. So when you can think about it in your own assay, what's the lowest signal I'm going to see? What's the highest? So what's my background and what's my signal? And what's the variance around those? And so what the Z prime factor is actually measuring is how far apart are these things? And so those values can range, obviously, if you look at the equation. They can go from less than 0 to 1, 1 being an ideal assay where you've got this sort of this infinite gap between. They're very tight. But really what we're trying to find is something that's in the range of probably about 0.5 or greater for most of the assays, and that's what we're trying to work on. So really what it means is how far can we get your signal from your background and how tight can we get the data. Those are the factors that you can see play a big role in here. So, obviously the bigger we can get that gap, then this number gets very small and this stays closer to 1, or if we can keep this number very small, obviously we can go that way. So this is an example of a cell-based assay. It's a 2D cell monolayer. We did growth curves on these cells with and without DMSO. All the drugs that we use in our screens are in DMSO. So we always try to evaluate to make sure that the DMSO concentration that we're screening at doesn't have an effect on the growth rate of the cells. And so it's us trying to find out how many cells to put in the well to actually do this growth curve. And you can see how the cells look. These are DAPI stained nuclei, and so this is the day 0 cells. These is after 3 days of growth. And you can see the distribution between the two. So you've got fairly tight values at 3 days and very tight at day 0 and a nice separation between the two. So if you look at these values, again, signal to background, signal to noise, it's the same data I just showed you, but our Z prime, the calculated, is about 0.6. So it's above 0.5. It's a pretty good assay. Every once in a while you get an outlier, but it's screening is what it is. So we were able to do a screen with these cell lines. This is a comparison of 2 different cell lines in response to a couple of different drugs. You can see that we have some similarities, we have some differences, and we were actually able to get a nice assay from these. Each of these is a triplet data point you can see. So you can see up here we've got some fairly tight data. It gets a little looser down in here. But that's really what we're trying to look for. But not everything goes the way you expect. Here's a cell line that didn't grow. So the green, again, is the day 0. Here's the day 3. They just sat there for 3 days and just really didn't do much of anything. Well, jumped ahead. See, you saw the end before I got there. Let me walk back. Okay. This is the second cell line that we did. And I don't know, does anybody notice anything about these graphs, these heat maps? Anything stand out to you at the moment before we go through the rest of it? I'm sorry? Speak up, I can hear.
It's a factor of 10.
Okay, well this is a growth curve again. So this is the day 0, this is the day after they plated, and you can see that the cells grew. So what I wanted to show you is in this one, so here, these are the same sort of parameters that we saw before. Average standard deviation, our CVs. The CV is a little high in this one, right? So this is all 384 values here. We had a signal to noise of 7 and a signal of 77, which sounds really good, right? But let's dig a little bit deeper. So as we look at the day 3 data, the first thing that started to stand out, if you look at those CVs, you can see this alternating pattern. And if you look at the heat map, you can actually start to see it starts to come out at you. And you can see it here. So this is some of the statistics that we look at when we're doing assay evaluation, is how does the data look? So what was going on? Then we saw this graph. Now does this graph look like any of the ones that we saw before? No, this one's definitely got a problem, doesn't it? So you can see, so if you look at the distances here, each one of these gaps is a row on the plate. So you can see in row A, there's this increase, and then row B is sort of normal, then row C, D, E, F, and so that it goes a lot with the pattern that we saw here. So if we dig a little bit deeper and we start looking at these values again, if I calculate my Z prime, now look where I am. And why is that? Because my variance is wide and the separation is more narrow. So you can see we had some issues with this. So this is a plate that we would reject. It doesn't meet the criteria. So rigor and reproducibility of the data is very important for us, so we rely a lot on the statistics. This is an example. So a lot of the experiments that we do, you're trying to inhibit something, but gain of function assays are really wonderful assays, and you can see why illustrated here. That in the absence of a signal that would cause signal generation, you've got pretty much background, but you can actually stimulate GFP production in these cells by modulation with an siRNA, and you can see over the period of time it grows, and we were able to actually calculate. So you're actually going from nothing to something, which is really a pretty nice assay. And those work out pretty well. How does it go for automation? So we took this assay, and this is sort of the automation scheme that we did. We were able to seed the cells and transfer the drugs, fixing and staining again for those cells, and then do the imaging and the analysis. And here you can see, as we increase drug concentration, you can see this nice gain of function. The DAPI stain here, not much change in the number of nuclei of the cells, so we didn't have much of a cytotoxic effect where we were actually able to stimulate, and then we can get full dose response curves in those. So this is an example of that kind of an assay, a reporter assay that really works pretty well. Again, rigor and reproducibility. So if we take that same assay and we start looking at numbers of plates, you can see for the most part it's fairly reproducible. And then we also run on-plate controls. We run dose response curves on every plate, and we look at each one of these curves as a plate, and they're all numbered down here. This is the same ones here. And you can see how tight the data is from that. And so that allows us to monitor from day to day how well the assay is performing. So we're always looking for those kinds of controls to include in the assay. So back to our plate. So once we've developed the assay, we've done some optimization, then we can actually do the screen. Again, we're trying to look for a simple assay that's robust and reproducible. And then really screening is removing things that aren't active. We're going to screen thousands of compounds and we're going to come up with a few things that are active. Well, what did we do? We threw away 99% of everything else. So we actually filtered out the things that aren't working. To validate those hits, then how are you going to see what's going on? So obviously false positives, false negatives. Doug touched on a few of these things in his luciferase presentation. You can actually inhibit luciferase. That's one of the problems, right? If you have a luciferase reporter, the compound you screened might actually inhibit the luciferase. So that would be a false negative, right? Or a false positive, I'm sorry. Chemical interference is another one, again, reporters. So then you want to try to do other screens to help you identify these. And if it's a single agent screen, you might want to run a dose response curve. If you ran a dose response curve, there are other things you can do. If you do orthogonal screens where you might look at a different endpoint or a different report of your endpoint. So you might have done a luciferase assay, but then you might want to do a reporter assay of some other kind. But the nice thing about orthogonal screens is that you're actually keeping things. These are things that actives you want to keep, so it's reinforcing that you've got target activity. The opposite of that would be a counter screen. So you're eliminating things from your active pool. That might be things that you may not want cytotoxic compounds. That would be an easy one to look at. You're doing a screen, you want to eliminate any compound that's toxic to your cells. So obviously you don't want to keep those. And then you need to check chemical identity and purity at some point. And then secondary screens, obviously these are the lower throughput screens, but more biologically relevant. They could be Westerns, RNA, there's a lot of things that you could plug in here. It also allows you to start to develop some structure-activity relationship, which is what you should see in your molecules. If it really is an active compound acting at that site, if you can, you should start to see analogs of that should behave similarly. For us, the approach we can take is analog by catalog, basically. We don't have chemistry support, so we're not going to be making compounds for you, but we'll go out to the commercial sources and see if we can find analogs that we may not already have, and we can explore them that way. And again, establishing biological plausibility is important. And as you noticed probably from that first presentation and from Doug's presentation, it takes a village pretty much to do the screening. And these are some of the technologies and techniques that are necessary to do that. And obviously the first thing is they're not all going to be available in one laboratory. So the question you might ask me is, how do I do that? And really the GCC is the way to do that, really. If you think about it, the Gulf Coast Consortia brings people who have an interest in drug discovery from all of these institutions in and around Houston. And they're formed into consortia, and there are many different consortia. Ours used to be called Chemical Genomics. It's now the Innovative Drug Discovery and Development Consortia. There are clusters. These are interest groups that may someday make it to being a consortium. And there are initiatives. I mean, Suzanne has worked really hard on this junior faculty network, and then obviously she mentioned earlier the shared equipment. And if you just look at our own consortium, there are shared cores. So we have combinatorial drug discovery, that's us. There's pharmacokinetics, that's Dong Liang at TSU. Mike Mancini and his group at Baylor, Advanced Microscopy and Image Informatics. And then our newest core, which you'll hear about in just a couple minutes, High-Throughput Flow Cytometry. It's a new area for us. It's an area we hadn't been able to explore. And these is a lot of the cores that CPRIT has funded. And they're not all GCC. The three in yellow are the ones that are at the IBT and Baylor. Mike's is actually a collaboration between IBT and Baylor College of Medicine. Dong Liang at TSU. You've got the DNA Encoded, which is coming up in another talk like this in a couple of months. That's Damien Young and Marty Matzik at Baylor. So you can see by networking through, and the nice thing about the GCC is it enables us to do that. There are no barriers between working with investigators from different places. Anybody from any institution in the GCC can come to my laboratory and we can start working together, and there's no paperwork. You don't have to do an MTA and a CDA and all the other things necessary just to start screening, unless you demand it. I mean, for your own protection, you can do that, but there's no barriers to do that, so the silos are broken down and gone, and that's one of the advantages of the GCC. This is just a map to show you where we are, just to locate you, Holcomb, Maine, and Pressler. The IBT is right here. So we're just on the outskirts of the Texas Medical Center, but we're well within walking distance. So it makes collaboration easy. We come to you, you can come to us, and it really works out very well for everybody. So one of the things that we've worked on, these are the different goals of projects that we've worked on: targeted identification, new probes for target validation, or just so you can do research with your molecule, with your protein of interest. Therapeutic vulnerabilities in new systems, and then identifying drugs and drug combinations for translational medicines. We work on small-scale projects. If you just need data to support a grant, we're more than happy to work with you to do that. We try to do simple endpoints as best we can. Those ones have the most rapid turnaround, but that's not limiting. We also are willing to work on large projects with large panels of cells. Combination screens always take a lot of work. Cellular models, if you've got something that's much more complex, or organisms, we've done a few zebrafish screens, so we can actually go to organisms if we need to, and we are also doing a lot more live cell and time-lapse imaging. Some of the projects that we've worked on, I always have to put Suzanne's in here. She did the dengue viral protease. Some of the other ones that we've worked on, these are more of the biochemical screens. These are projects where we actually have the core written into grants that have been funded. And then these are just other types of targets that we've worked on. And again, organoids is an area that we've been exploring for the last year and has really taken off. Co-culture systems, we have a few models in those, and 3D cultures such as spheroids. And tissue chips are coming along as well, so we have a small initiative in that as well. I'll get to the end. This is how we've developed over time. We started in 2006 at the University of Texas Health Science Center. And we were mostly doing synthetic lethal screens with siRNAs, and we were doing small molecule screens, and most of it was biochemical at the time. And you can sort of see our transition over the years, going more, less from biochemical to more imaging-based screens. We added organoid, humans on a chip. Here we added organoids in the last year or two, and spheroids. So the model systems are getting more involved, more tumor-like in the case of cancer, getting away from 2D monolayers into more organoid type structures. And then if CPRIT gets reauthorized, and this is my point to say, if you vote, please go out in November and vote positive for CPRIT. It's up for renewal this year. It's been a great benefit for us. You can see that we've been funded by CPRIT since 2011. It's the way we've been able to sustain the core. And really what that grant's going to try to focus on, primarily organoids, tissue chips, biosensors, and metabolism. So that's sort of moving in that same direction. This is just some of the automation that we have in the lab. Like I said, we don't have any big yellow arms, but we do have automated stations where we can do everything from pipettes to pin tools and acoustic transfer. We have readout devices that can look at just about most any of the assay formats that might be common for biochemical assays. And then our imaging platforms, we have an in-cell that's mostly for fixed cells, and then our live cell imaging is done on the molecular devices platforms. And then we have a lot of data analysis on the back end. You can imagine we generate an awful lot of data. And being able to store that data, and we have redundant storage both in Houston and College Station. Since Houston's in a hurricane zone, we figured the safest thing to do is to mirror our data in 2 different sites, so we have some security in our data and a lot of infrastructure. At this point, I'd like to introduce Margie Menchgamba, who heads our flow cytometry program. This is sort of a schematic of what we think we're going to be putting in place, and you can see, similar to what Doug had showed earlier in one of his slides, you've got some basic robotics and automation, cell culture incubator, manipulators, and then a readout device, and in this case, the readout device is a flow cytometer that allows us to feed it plates automatically. So I'm going to introduce Margie some of her slides in my time, and then we'll finish up.
So hi, you guys. For those of you who don't know me, my name is Margie Menchgamba. I'm just down the street with Cliff. Of course, this is the picture of the IBT, and as you can see, I'm the new director of the new high-throughput flow cytometry core facility. My background is in immune cell biology, which is why I do flow cytometry, and obviously you've already heard Cliff's background. Well, Cliff already showed you that picture, so you guys know where we are. We're just down the street on the corner of Pressler and Holcomb. But the first program I want to tell you about, because we got funded, right? So we have money to do stuff with. And so one of the things we want to do is to offer this pilot grant program. And this is for, so our goal is to fund, to offer pilot funds to innovative applications that want to use our high-throughput flow cytometry instrument that Cliff just showed you. But it has to be related to cancer research, because this is, our focus of the grant is immuno-oncology. So if you can somehow maneuver your project to be immuno-oncology, you'll stand a chance in the competition. A little more details about the awards program. So these are going to be 3 awards, $25,000 a year, and they're individual grants. The restriction, though, is that you need to have an institutional match. Okay, so your institution needs to match the $25,000, but that gives you $50,000 to troubleshoot and to pilot, a project that you're interested in. There are some restrictions. This is for early stage investigators, established investigators, and also postdocs. So postdocs, if you guys are toward the end of your career where you want to get some preliminary data, you guys should apply. Now, all GCC institutions are eligible, so everybody here can apply. It'll be administered by us. And the awardees will be selected by the scientific advisory committee that we're putting together right now. So that's the first thing. Oh, and the scoring criteria is this. So it has to, I mentioned, it has to have relevance to cancer therapeutics. It has to be the significance and impact, obviously, and then the quality of the research plan. So that's how the applications will be scored. And the other program that we have is the Post-doctoral mentoring program. So the goal for this one is to prepare young scientists who are interested in careers in academic or industrial drug discovery research. And this is in partnership with the GCC IDDD, apparently. I didn't update this slide. Well, I didn't know, okay. All right, so what are some of the activities we have planned for this? Well, mainly, we are planning on offering mentored rotations in our core facilities. Cliff is the core director of drug discovery, and I'm the flow cytometry core director. So obviously, we're just merging our core facilities together, so you'd be able to rotate through both. We will offer roundtable discussions that, such as this one with specialized technologies, instrumentation, again, all related to cancer therapeutics research. And also participation in workshops such as this. So those of you who are interested, information will come out. We haven't received the money just yet, but when it does, when we get it, we will start making announcements and send through Suzanne, and you guys can plan accordingly. And I think that's it. Yeah, well, Cliff asked me to mention this conference in May, but Suzanne already did, so no need for that.
If you look at all the different cell lines and all the different classes and many of the targets that we were looking at, a couple of them started to fall out as being outliers from the rest. The aurora kinase, the HDACs, and then PLK1 and proteasome inhibitors. And so that was the extent of the screening that we did. And then their group went on and did modeling, and then they did the secondary assays. And that's what I'm showing here, because remember, the secondary assays are important to prove the biology. So the original hypothesis was that there would be a difference between HPV positive and negative cells, and that actually didn't turn out to be the case. So that wasn't in the data. It wasn't supported by that. But they went on to find that there were certain biomarkers in these cells, and if you looked at the gene profiles, they actually found some drivers in those cells that could be targeted by drugs, and one of those drugs then was an aurora kinase inhibitor. And you can see if you look at apoptotic responsive cells, with and without the drugs, in different cell lines, with and without the drug, you can see an increase in apoptotic cells. If you look at tumor size in an animal, you can see that there's a large reduction in the growth of the size of the tumor. And you can actually look at protein expression in cleaved Cas space. So they had a number of secondary screens that they were looking to make sure that the compounds were working in the way they thought they would. And from this, there were several conclusions that came from that. First of all, this was, as far as they are aware, this is the largest reported pharmacologic profiling of HPV positive and negative head and neck squamous cell and cervical cell lines. So it's an important bulk of information. But they identified the aurora kinase inhibitor sensitivity, which was important. And it seemed to align with the lysine methyltransferase D2 mutation in these cells. And so the data is now supporting a phase 1 clinical trial in these patients. And so we're actually translating the data that we generated in the screen, and it's moving towards clinical trial. The second project that we're working on, spending a good deal of time on, is a multi-investigator award with MD Anderson in triple-negative breast cancer. And this one's actually tied to a clinical trial, and that's what's the interesting part of this. Is the cells that we're actually screening are coming from patient biopsies in the form of patient-derived xenografts. And so before a patient starts the treatment protocol standard of care, a pre-treatment biopsy is taken from that patient. And we test all of those cell lines, so every patient is getting biopsied. And then they'll go through standard of care. And that's usually 4 cycles of AC. And then at that point, there's a decision to be made. Did the patient respond to the treatment or not? And if the patient didn't respond, well, anyway, we're doing a biopsy, we're doing a biopsy here if there is residual disease. So if there's something to biopsy, if the patient didn't respond or didn't respond fully, they're taking another biopsy, and that goes through the process of going through PDXs and then screening as well. The idea is, can you identify therapies that this patient then might be able to go and be treated with? And so that's where this bifurcation comes in. The standard is to do taxane therapy. That's standard of care, but that doesn't work for all patients. It only works for about 50%. What about the other 50%? So the idea was, can you then do a targeted therapy? By doing the screening first, identifying potential targets, can you take those women that may not be a good candidate for taxanes and find another way to go? And then if there is still residual disease, again, after this treatment, then we get those cells as well to try to identify where the resistance is coming from. So it's an adaptive clinical trial. It has many arms involved with it. But the screening is actually trying to find ways to either use combination therapy or single agent therapy to try to treat patients that might be resistant to treatment otherwise. And for this screen, we're using approved oncology drugs from the NCI. So it's a set of drugs that we have there, and you can see the overlap between the two. And illustrating them was a challenge for us. You know, looking at those cluster maps is a good way of doing it, but it doesn't tell you very much. So we came up with this visualization, which we're calling a pharmacologic tree. And so what we've done is you do the associations between targets and pathways, and you can see how they illustrate here. And then we are able to look at them that way. And the reason we did that is when you start comparing different cell lines. And so here we've got a patient a PDX from a MET, from a patient. And then pre-treatment A, so this is before treatment for patient A, a different patient, patient B, patient C. And you can see, I just highlighted some areas where you can see some very noticeable differences between. And one of them that's interesting is this one. This is the taxanes area, so microtubules and taxanes. So this particular patient is very susceptible to taxanes. So maybe this patient might do better being seeing a taxane before they've even had standard of care. It's something to think about. Whereas these other ones are fairly resistant to it. And then this is a mid-treatment patient. So this is the pre-treatment for the patient. This is their post-treatment. You can see there's just some not much difference between them. A little bit difference up here, which is in our HDACs, but there's not a lot of difference between these two. And so we're starting to get more and more. This is all that we've really gotten so far, but we're starting to get more of these matched samples, and that's really what our part of that grant is to study these and come up with rational drug design. So how do we do that? There's a couple of ways to do drug combinations. The first is just functional observation. Can we take the information that we're getting from the drug sensitivity and come up with ways to do that? And there's two ways to do that. You can just do pairwise combinations. You take all the active compounds and just brute force put them together and see how they respond. The other is this anchor probe approach, where the anchor might be a standard of care or might be a driver mutation, and then we can actually probe with active compounds from here to see if there are multiple pathways that might be involved, that might be active for that patient. Or we can do what we call the integrative approach, because this grant also has a lot of omics data being profiled as well. So all of these patients are also being characterized for all of their genomic profiles. And so can we actually merge that data with the drug profiling data in a way that we can actually come up with combinations? And again, that's the whole purpose for this grant for us, is to try to do the integration, trying to do prediction to drive clinical validity of the screen and then reversing resistance in these patients. So that's what we're trying to do. Where are we going next? In the last couple of slides, the last couple of minutes, just other areas that we're moving into. And again, this is looking at the advanced cellular models. So again, looking at as the area moves forward, 3D cultures and gels, organoids and spheroids. And so this is an example of a multicellular spheroid assay where the cells are plated and they'll self-associate into a spheroid. And then we're able to monitor over days, since we do repeat imaging. So the microscope's attached to a tissue culture incubator, and we're able to just continuously read those back and forth. And you can see that some cell lines will form nice spheroids, others will not. So that's part of the assay development phase that we talked about. And you can see that's reflected here of the different cell lines, different cancers that we've been looking at, cell lines, and you can see sort of the success rate of some of those. Overall, about 50% of the cells that we do will actually form some kind of a structure that will show growth over time. Reproducibility and robust reproducibility are obviously important here, and so for the screen that we're doing, we typically do a 3 concentration screen. We do 3 or 4 replicates per plate. That's where we're able to get pretty good Z-scores on those. The size of the screens we can do is somewhere in the range of up to 30 plates in a day, and we can do that probably twice a week. So we can do quite a lot of screening in this way. Organoids is the other area that we've been moving into, and you can see that the cells are plated as single cells and then allowed to grow into these organoids over time. And so this is a sort of a growth. You can see the growth over time. The cells labeled here, and this is sort of the profiles that we're trying to do. So you can see that we allow them to grow a couple of days. So we actually let them grow to make sure that they're actually starting to form a spheroid before we even do the drug addition. And then we let them go again beyond that. So we're trying to allow the cells to go through at least 2 or 3 cell cycles, so that if the DNA damage is necessary for the effect, that it gives them an opportunity to actually see the effect. And then we're doing a small Z-stack and then best focal projections to get these kinds of images. And then we also follow that with a viability assay. And then deep neural net analysis of the data of the bright field images. And these are just some of the different kinds of spheroids that you can see. We get nice ones that form nice little organoids here, but then we've got these ones that actually have some morphology to them. You can see that they've sent out processes and are actually making like a spiky organoid. So this is just a small screen that we did. There's 4 replicates. You can see just a series of time over time in the cells here. And then, so how do we do the analysis? So up until now, the standard for image analysis was doing image segmentation and then extracting features and then trying to do feature analysis, and you can see this is sort of a profile of a drug screen that we did, and this was single cell analysis, so we were able to easily segment the images. So you can think of a 2D monolayer. If you think back to my image where I had the DAPI stained nuclei, it's easy to sit there and count nuclei. They're single spots, and they're easy to do. Think about those organoids, they're a little bit more of a challenge. So we're trying to develop other ways to do that, and one of the ways to do that is to use some of the machine learning approaches to do feature extraction, and then you can still do unsupervised learning with the principal component analysis. You can see that it recapitulates the single cell analysis fairly well. What if you get even more abstract and actually start to use the deep neural net analysis? So pretty much a black box approach to doing the analysis of the features. You can see we're still able to do fairly good representation. So then the question we had was, can you actually apply that to spheroids? And so the next question was, if we took a spheroid screen, we're doing a number of compounds and increasing concentrations. So from right to left, increasing concentrations of drug. This is cell titer glow, so we're looking at viability. So as you increase in concentration, you're seeing the cell viability go down. The question we asked was, could we actually, without doing anything else than analyzing these bright field images with the machine learning, could we actually get similar profiles? And you can see that we see portability. And so that's the approach that we're in right now, and this is where we're trying to develop analysis algorithms to do this kind of work. We could take those and then actually generate the dose response curves, and the dose response between the cell titer glow and the machine learning were spot on. And then lastly, just some other initiatives that we're working in. So another area that we've started working in is tissue chips. This was an NCATS funded program that we've been involved with now for a couple of years. I just put a couple of examples of some of the chips we've worked on. This is a heart here. This is a salivary gland. You might not have noticed it. It's about this a little bit smaller than a dime. This is a blood vessel over here. So you've got 4 different blood vessels that are formed in here. This is a kidney. And this is synthetic skin, or skin that was actually grown in a dish. And we're trying to do profiling and testing, toxicity testing, drug testing in models that are more human-like and in model systems that might be amenable to more easily for drug screening. Another is integration of biosensors. Can we actually take, in this case, our idea here is to take the seahorse concept, where you can actually look at glucose or glycolysis oxidative phosphorylation and extrapolate that into 3D models. And so we're working with Agilent with some of their soluble biomarkers and biosensors now and trying to develop those into a plate-based system. Momentous has a plate that has microchannels in it that allows you to seed cells and allows the cells to grow. So it gives us an opportunity to actually to do those kinds of work in a gel-based system. So this is the 3D models. And then with Agilent's biosensors, they can be integrated into the gel, and so we'll be actually looking to see if we can measure changes in pH, if we can look at changes in glucose and oxygen. The system for that is an image-based system. Again, it's an Image Express with a plate manipulator and an incubator, but the incubator is rather unique in that in the momentous plates it has a channel for microflow, and so the shelves in the incubator actually rock back and forth. So you can, as you tilt the plate down, you can get gravity flow and you can generate flow within the chip. So if you have a model system that needs flow, we can actually generate model microflow in those as well. And then lastly, TMC3, people are starting to hear about the initiative there, where you actually have a chance for public-private partnerships to form. Each of the institutions in the medical center is going to be participating. Whoops, wrong button. In the screening, well, heck, technical error, user failure. So the core is slated, our core is slated to be the high throughput screening core there as well. So it's not like our access is going to go away with TMC3. We're actually hoping to expand that into opportunities perhaps with biopharma or pharma in general as they start to come into this program as well. The proximity will be working there in our favor. And so really the whole initiative of TMC, which is the TMC, the 3 they call Third Coast. So as Doug pointed out, a lot of the screening happens either on the East Coast or the West Coast. Well, Texas wants to be the Third Coast, so we want that screening to come here as well, and drug discovery. The folks that do the work in the lab, Peter Davis is the director of our center and also head of the core, myself. Mary, Goen, Ivy, and Young are the screeners. These are the professionals that work in the lab. They're assay development experts, and they've been automation experts as well. Kurt Zong actually is our rigor and reproducibility guru. He runs a core facility in rigor and reproducibility. Leoncio helps us with the microscopy and the imaging. Ken Lee is a graduate student that's finishing up but has been involved in a lot of the pediatric high-grade glioma, the work that we've been doing. Reed Powell and Pankaj Singh are the informatics guys, so they're the ones actually doing the deep learning model generation. And Lego has been involved with the TNBC project for us. And I'd like to finish with this, the assay guidance manual. As Doug pointed out, it is the Bible. And really, if you want to learn to do screening and they cover so many different topics, no matter what your model system is, there's a good chance you'll find something close to it in this book. And you can see one of the editors is with us today. But I refer you to it. It's just Google Assay Guidance Manual. It's the first hit that comes up. I encourage you to go through it. And that's my last slide. So thank you.